I'm going to get the next speaker up. Jorik is going to talk. He's another speaker from Netherlands, and he will be talking about biomarkers and metabolomics.
Thank you very much. Let's see if this works. All right, good day everyone. My name is Jorik, and currently I'm doing my PhD in the group of Stefan Kemp and Mark Engelen in Amsterdam UMC, and my research focuses on the use of metabolomics. Sorry, I can take this too. Sorry. Yeah, it's okay. So my research is focused on the use of lipidomics and metabolomics, specifically using mass spec to study adrenoleukodystrophy. And we use these methods to actually identify new biomarkers that hopefully give us some information that is relevant for things like disease progression, but also the different phenotypes of ALD. Now, traditionally, when we look at biomarkers in ALD, as you all know, ALD is characterized by an accumulation of very long-chain fatty acids, and here you can actually see an example of one of those very long-chain fatty acids. So this is a fatty acid head group connected to a carbon chain, and as you can see, the carbon chain is quite long, hence the name very long-chain fatty acid. You can measure this fatty acid with GCMS, and that's actually what you see right here. So this is you can use this fatty acid as a diagnostic marker, with here on the y-axis the concentration of this fatty acid, and on the x-axis various groups. When you actually compare healthy controls to ALD men, you can see that there's quite a big difference in this in the level of this fatty acid. However, when you consider ALD women, there's some significant overlap between patients and healthy controls. That's why the field has been looking for new biomarkers, and one marker that's been used quite a lot at the moment is this one, and so this is an LPC26O marker, and it looks a bit like this fatty acid. However, this fatty acid is now incorporated into a more complex lipid structure. It's connected to a glycerol backbone here that is connected to phosphatidylcholine head group. And now, when we consider the levels in controls and patients, again here on the concentration on the y-axis and various groups on the x-axis, we can see again a large difference between controls and ALD males, and this time also a more bigger difference in ALD women compared to healthy controls. So in terms of the sensitivity of this marker, this is quite a better option compared to the traditional C26 fatty acid marker. Also, when you consider the methods for analyzing these markers, this is done with GCMS. It's quite a complicated, long method, whereas this you can measure with directly with mass spec or in combination with liquid chromatography. And because this method is a lot easier, this marker is also used in the newborn screening program in the Netherlands. Now, we actually want to move from this fatty acid marker to this LPC marker. We want to replace this method for the LPC method. And that's why in our lab we started measuring samples using both of these methods. And that's actually being displayed here. So here on the y-axis you can see the concentration of the fatty acid marker. And here on the x-axis you can see the concentration of the LPC marker. And these areas, they refer to the normal range for healthy controls. And when we compare these methods, so all these dots are the same. So this is one sample measured on both methods. Here on the top right, we can actually see that these are samples that are significantly increased in both methods. So these are ALD patients, Zellweger patients. And here you can actually see that there are several controls that fall in the normal range. But of course the most interesting ones are these red dots, because these represent markers that are elevated in one method but fall in the normal range with the other method. So there's a certain discrepancy there. And actually when you look at the ratio of C26 to C22 in these samples right here, they normalize. So this goes to show that also this, when you consider this traditional marker in ALD, you also have to consider the ratio of the C26 to C22. And these markers right here, so these are normal in the fatty acid method, but elevated in the LPC method. And we're actually currently investigating what the clinical outcome of these patients was. So yeah, that will be quite interesting. Now, these methods are very valuable. They can be used for diagnostic purposes. However, they don't provide any information in terms of disease progression or the various subtypes in ALD. So when we consider the current situation in males, this is what we have. We have a group of male patients that are diagnosed with ALD. A certain group will develop cerebral ALD. A certain group will not develop this cerebral form and go on and develop the spinal cord. And of course, there's also some patients that at a later stage make the transition from the spinal cord subgroup to the cerebral form. Yeah, and currently we don't know which group will develop which phenotype. There's also a large variety in disease progression in terms of the myelopathy. And currently we can't predict, we don't know what causes this large variety. So one of our goals is to actually develop methods, new methods that can characterize the metabolic changes that happen in all these subgroups. By characterizing these metabolic changes, we hope to find markers or profiles that are predictive for the various subtypes in ALD, but also are markers for things like progression of disease, that are able to quantify the degree of change that's happening over time, but also things like therapy efficiency. And of course, when we characterize these metabolic changes, we also want to look at, can we identify certain risk factors for the development of the cerebral form, and hopefully leading to early diagnosis of cerebral ALD. Now we use a metabolomics and lipidomics approach for this. And metabolomics or lipidomics is the use of analytical technology to profile and quantify metabolites on a large scale. We do this because we want to characterize certain biological states. And by characterizing these biological states in different contexts, so for example in different subgroups, we hope to extract meaningful information that are clinically relevant. Now, metabolites, they are the result of metabolism within the biological system. They're relatively small compared to other biomolecules. Here you can actually see some examples of various metabolites. What immediately becomes clear is that there's a large variety in terms of chemical structure. And this large variety also means that there's a large variety in chemical properties of these compounds. Also, these metabolites, they exist within the biological system in large concentration ranges. So some metabolites are very low abundant, whereas others are very high abundant. They also exist in different matrices. Think of muscle tissue, blood, urine, all quite different. And then lastly, there are quite a lot of them. So HMDB, that's an online metabolite database, currently holds over 114,000 metabolites. So all these factors combined result in the fact that the simultaneous analysis of these compounds is actually technically quite challenging. Now, in terms of how we approach this at our lab, we start with a certain sample, and the sample can be anything that contains biomolecules, both from model animals and patient's material. And of course, the obvious ones are blood, spinal fluids, but also brain tissue, cell systems. And what's interesting in our research is that we follow a large group of patients over time. And what's interesting is that this is around 80 males, and there are various phenotypes within these groups. So we can actually divide this cohort up into different subtypes and look at the differences there. So we take these samples and then we extract them from their biological matrix, and we use a very famous extraction method for this called Blind Dyer. This is an extraction method originally published in the 50s, so quite old, but it works like a charm. It basically uses chloroform, methanol, and water to create a two-phase system, and you can compare this with this glass right here. As you can see, a fat layer and a water layer, they don't mix. So an organic and a water layer, they don't mix. They result in a two-phase system, and that's also what we do in our sample preparation. So we use chloroform, water, and methanol to create an organic and a water phase. And the water phase or the polar phase, they contain all the polar metabolites, and the apolar phase or the organic phase, they contain all the apolar metabolites. And these apolar metabolites we also call lipids. Now then we take this apolar layer that contains all the lipids and we perform analysis on this, and generally we call this lipidomics, so metabolomics of lipids is also called lipidomics. Our lipidomics profile utilizes LC-MS, so we use liquid chromatography to reduce sample complexity. And chromatography is nothing else than the chemical separation of certain lipids to reduce this sample complexity. So analysis is more efficient. And we use two chemical separations for this, called reverse phase and normal phase. And they relate to the chemical nature of the separation. And that chromatography you can compare with a tube, and the tube is coated on the inside with a certain chemical coating, and there's a liquid going through it. And when you introduce a sample in this tube, certain compounds within your sample have different speeds where if they go to the tube. So this different speeds actually results in separation. After separation, they will go to the mass spectrometer, and mass spectrometry is actually nothing else than a very sophisticated molecular scale. So it measures the mass of certain molecules. And here you see three examples of this. So these are quite different molecules. And by measuring very precisely their mass, we can relate this mass to the chemical structure, and thereby identify these compounds. We do this both in positive and negative mode, and this relates to the charge that molecules can have. Certain molecules are ionized in positive mode, others in negative mode. So by combining, we can increase the coverage. Recently we also started to do metabolomics. So we take the polar layer, the water layer, and we also perform LC-MS on this. And we use a system called HILIC for this, and this is specifically for polar metabolites and also in positive and negative mode. Now these measurements and these experiments, they generate a lot of data, and you need sophisticated data analysis strategies to extract as much information as possible, and we have a dedicated bioinformatics pipeline to do this. Now what's important to note is that both of these analyses, they operate in full scan mode, meaning given a certain mass range, they measure all masses. This is a form of untargeted analysis. And then we take these untargeted analysis, and it actually contains over 20,000 features. And of these 20,000 features, we can identify over 1,300 lipids and around 200 metabolites. What this basically shows is that there's still room for extra identification. More than, let's say 90% of the features we currently don't identify. But if we in the future identify more compounds, we can actually go back in our data, and because we measure in targeted data, you can actually identify new compounds within this data. Now we also develop new technology, and this is the machine that we use for this. So this is called a timsTOF, and this machine actually combines different separation mechanisms. It does mass spectrometry, so it determines the mass of molecules, but it also does ion mobility, which is a technique that is very complementary to MS, and we use this to identify new compounds and also to develop new methods to analyze those compounds. And there's a constant feedback loop between this development unit and our existing platforms. Now when we consider our lipidomics pipeline and we apply this to ALD samples, looks something like this. So this is lipidomics analysis on ALD plasma, in this case males, where we compare 12 controls versus 12 AMN patients. And that is plotted in a volcano plot. So a volcano plot plots the fold change versus the significance, meaning here in the top right, these are all compounds that are significantly increased in ALD patients. Now when we zoom in, we can actually see that these are multiple lipid species. So the first two letters, they're an abbreviation of the subfamily of lipids, and the numbers that follow behind it, they give some information on the total length of the lipids. Now as you can see, there are various subfamilies that are changing in ALD, and it really indicates that in ALD patients there's a systemic change in their lipid profile. Now we can also express those results in PCA analysis, and you can clearly see that we can distinguish these two groups. And also in a heat map you really see that there are large systemic changes that happen in ALD patients. We can also do this in the cell system. So this is in fibroblast, where we compare 5 controls versus 11 ALD patients, and again systemic change is happening there. But also in women we can perform this analysis, and what's interesting is that women, as you can see, have less significantly changing lipids, but still we see lipids that of various subfamilies are changing. So this also shows you that even or that also women have systemic changes in their lipid profile. And we can take this data and extract biomarker candidates from this. So remember that C26 fatty acid, it showed some overlap between controls and ALD women. Well, we can take our lipidomics data and we can identify some candidate biomarkers. So these are all biomarkers that are able to distinguish ALD women from controls. And moreover, these are all ratios of certain lipid species. We can also do this in tissue and model animals. So this is brain tissue of ALD knockout mice. Again, you can see how many changes we can detect. Now this is interesting. We can see that there are quite some changes that happen as a result of ALD compared to healthy controls. But what about subgroups? Well, there's a large variety in ALD progression, of course, and when we take a closer look at various subgroups, we see pictures like this. So this is AMN patients versus cerebral patients, and you can actually see that these patients are quite similar to each other, or at least far more similar than the other picture that I showed. And these are 47 AMN patients versus 7 cerebral patients. But still we can see various lipids that are changing. And when we go a step further, so this is cerebral patients where we compare a normal cerebral patient that have progression cerebral lesions versus cerebral patients where the lesions are halted or arrested. And even in these two groups we can find significant differences in their lipid profile. Now there are quite some interesting lipids that are changing here. One of the interesting ones is this one. So this is a phosphatidyl inositol. It looks like this. So it's composed of 2 fatty acids connected to a glycerol backbone that's connected to an inositol head group. And this marker is actually interesting because as you can see here in the box plot, we can distinguish cerebral patients where that are progressive in nature, or that or cerebral patients that have arrested lesions. And also here there's another one. There's actually a whole panel of these phosphatidyl inositols, and these inositols are interesting because they are involved in many essential metabolic processes, including building blocks of membranes, but also things like cell signaling. And what's interesting is that these inositols are especially abundant in brain tissue. Around 10 to 15% of total phospholipids in the brain is composed of inositols. And these inositols can also be phosphorylated, so that means that certain phosphate groups are attached to this head group. And these phosphorylated forms are associated with other neurological diseases, including Parkinson's and Alzheimer's. Like I said, we also do metabolomics, and also here we can see some changes that are happening between the progressive form of the cerebral and the arrested form of cerebral ALD. Now, we also do research on new technology to improve our analysis. And one of these technologies is combining eye mobility with mass spectrometry. And that is shown here. So this is a map of our current machine, and this is your normal mass spectrometer. And by placing an eye mobility unit in front of it, we can perform analysis in sequence. And eye mobility is complementary to mass spectrometry. Remember, in mass spectrometry we determine the mass of compounds. However, there are various metabolite species that have the same mass but are actually different in chemical structure. And here you see two examples of this. So these are molecules that have the same mass but clearly have different chemical structures. Their fatty acid tails in this case have different lengths. And we are actually able to separate these compounds based on their mobility. And mobility, you can compare this with how aerodynamic you are when you're sitting on a bicycle. So these are different bicyclists, and they either sit up or they sit down. And if you go biking with a strong wind, you will really notice that you go with different speeds depending if you sit up or down. This is actually the same principle that we use for eye mobility analysis. How we separate them based on their steric structure. And by using this technology, we can make quite sophisticated biomolecular profiles of ALD samples. So here we have mass versus time. So this is the mass of our measurement, and this is the time that it takes for the analysis. And all these clusters, they are associated with different subfamilies of lipids. Now this is only mass. We can also do this with mobility. And when we consider our mobility measurement, we also see various clusters. And these clusters are also associated with different lipid subfamilies. And here we can actually correlate mass and mobility, and that's what you see here. So here we have mass on the y-axis and mobility on the x-axis. And you see that these two measurements are correlated, which makes sense. If molecules tend to increase in mass, so they get bigger, they tend to also increase in mobility. However, the most interesting molecules are on this horizontal line here, because these are molecules that have the same mass, so you are not able to separate or analyze them with mass spectrometry alone, but they do have different mobility values. So by making these very detailed biomolecular profiles of plasma or of ALD samples, we are able to compare those or relate those to different subgroups, and we are planning to also apply this new technology to that ALD cohort that I just showed. Now, concluding, I hope I convinced you that metabolomics and lipidomics are really powerful tools for studying ALD. Moreover, we need to validate these markers in external sample sets. We need to confirm if these markers that we just identified also hold up in external samples. In terms of the arrested versus the progressive form of cerebral ALD, we also want to measure these biomarkers in samples before and after the occurrence of these lesions, and see if the levels of these are associated with the occurrence of these lesions. And then finally, we also develop new technology. We combine eye mobility with mass spectrometry to really improve and get more detailed biomolecular profiles of ALD samples. And This is my acknowledgement site. This is the ALD group in the AMC and the tandem MS group in the AMC. And we're now also on Twitter if you want to keep updated with our research at ALD team. And we're also on Instagram, team_ALD. So with that, I would thank you for your attention.
A couple questions. And then... Okay, go ahead. Oh,
Hi, thank you, Eric. That's really impressive work. I have 3 super quick questions. Number 1, the identification of those biomarkers is really promising. How do you look at how they perform over time? Because how they perform over time is not how they perform cross-sectionally. That's number 1. Number 2, since you have such a robust metabolics and clinical data set, have you done some regression modeling against their EDSS or some form of global disease severity? And number 3, we were chuckling when we heard the inositol story because we've been talking about this in our lab and Anne's talking to us about this. One of the things we're looking into in these inositol species is that there is some difficulty in isolating them because of where they're phosphorylated. So there's, a lot of us know PIP3 that's a messenger for a whole host of things, but there may be different species. And then, so a 2,4 phosphorylated would have the same mass as a 3,5 phosphorylated. With your ion mobility, can you differentiate between them? Thanks.
Yeah, to answer your last question, that's exactly the plan that we're doing now. The thing is that these phosphorylated species are far more low abundant than the other inositols, but we really hope that we are able to separate these with ion mobility. In terms of looking at these biomarkers over time, we're following an ALD cohort, so we take samples every few months. And by comparing these samples, we hope to get information in terms of progression of these markers and see if they actually relate to their abundance. Your second question,
regression against some clinical markers?
Yeah, we're still doing that. It's actually quite new data that we have here, and it's one of the things that we're working on now to do some analysis in that regard. Yeah.
That would be really interesting to see.
Yeah,
Thank you so much.
I am curious whether you had the chance to compare the same sample run both by LCMS with Orbitrap and ion mobility with time of flight mass spectrometer, because with ion mobility you can gain some capabilities, but usually it comes with sensitivity penalty, and then resolution and mass accuracy of these instruments are also very different. So if you had a chance to compare your coverage, that would be
true. That's actually one of the projects that we're working on at the moment, to compare our Orbitrap to our timsTOF. In terms of the sensitivity loss, what's nice about this instrument is that it uses a new form of ion mobility called trapped ion mobility. And it basically means that the ion mobility cell is composed of 2 segments, which means that that you hardly don't have any loss of ions, because there are two segments to it. There's one segment that accumulates ions, and there's one segment that separates the ions. So while it's separating ions, it's also accumulating, and that way you have a duty cycle of almost 100%. So the sensitivity loss is far less than your traditional ion mobility methods. Now, in terms of your identification, yes, resolution is lower compared to an Orbitrap. However, what you do have with a QTOF is that it scans faster, so it's more sensitive, and it also has better isotopic patterns. And when you consider isotopic patterns, you can actually make up for the loss of resolution.
Are you using isotopic pattern to distinguish between second isotope feature and unsaturation?
You can use isotopic patterns to get information on your chemical species. And of course you have to correct for overlapping peaks, of course, because that's a problem with when you consider this. But when you combine it with ion mobility, you actually decrease the overlap in the isotopic pattern. And,
that's true. Mm-hmm.
So you have cleaner MS and MS-MS spectra because of it.
Thank you.